Good morning students. Welcome back to other session of our video. Here in the previous video what I have explained, once we'll have a revision, then what are the today's topics are going to be covered, that I'm going to explain, then we'll start our session. So here what are the topics we have completed previously? Here we have completed about the great scientists who contributed, and we have learned the Robert Hooke experiment, in what way he did an experiment, and similar experiment of observing the cells of matchstick. What activities we have completed? Observing cells in matchstick. Then here in the second, observing cells in the onion peel. Then onion peel. Third one, observing the cells in our cheek. Cells in the cheek. So here we completed the theoretical part and experimental part also. In the previous video, I have showed the how I have completed how to observe the cells of the onion peel. So now here, when again I will do the experiments, so then I will continue this one also. So now in this video, I am going to tell about the significant observation of about some scientists, and the significant observation especially on nucleus. Who are the scientists who observed and who given their significant contribution over the nucleus, we will see, and who discovered the nucleus and who given the name nucleus, and which type of cells he observed. And next we are going to do a theoretical part of the activity of observing the human nucleus in the onion peel, then observing the human cheek cells, that is in human cheek cells we are observing the nucleus, and then we are going to compare the nucleus, that is the position of the nucleus in the onion peel and the position of the cheek cell, and some differences you are observing while I am explaining. Please everyone listen carefully. Wherever I will explain, I want to show you a comparison between these two cells. So these are the topics are going to be covered in these videos. So everyone, are you ready? So let us start our session with a great. Now here I want to tell, I said no, significant observation. Significant means here it is a milestone. Means here you know that our Robert Hooke, understand, he discovered the cell. He discovered the cell, and when he discovered in 16th century, he given the name cell. After that means 17th, 18th century, this means at least 150, 180 years, then they discovered about a integral part of the cell named as nucleus. Understand? Here in our topic we are going to learn who are the scientists who discovered and who observed these integral part of the cell. Why? Because in the previous video I have shown the diagram also and I have given the name. This is the cell membrane, cytoplasm, nucleus. But nucleus word was first said by Robert Brown. Let us start this one. So here Robert Brown and Felix Fontana. See, he is the first person who first saw the nucleus. Saw, please everyone focus on the word. He first saw the nucleus in the epithelial cell. What is meant by epithelial cell? Here this is the outer layer of the body. Means it's the outer layer of the body of different tissues. So here if you see he found these nucleus in the first time by Felix Fontana. So here he saw, but he didn't given the name, and he didn't said that it is an integral, a very important part in the cell. He didn't give anything, just he observed in the epithelial cells. Then later in 18th century, the Robert Hooke, he saw different kinds, that is, he observed in different kinds of cells, especially the cells of orchid leaves, and when he observed a dark, a circular spot, more opaque. When you compare, you can see very clearly that one. Then, and he used the word integral part of the cell. Integral, most important part of the cell. Understand? He used the word integral part of the cell, and he was very anxious to see that whether these type of structure is present in all other different cells. Yes, when he observed different kinds of cell, he observed that this type of organelle is present. That means here the word nucleus was said by Robert Brown in 1831. That means in 16th century the word cell was said by Robert Hooke. Later, that is 150-180 years later, Robert Brown, by his contribution, by observing, and he said that in the cell, especially a circular spot, very opaque, and he given the name nucleus in the year 1831. See, Nana, in biology especially, you should have an awareness about the years and what is in which scientist, which year, and what he discovered. So this is a very, very important, especially in the CCE model. Understand? Here, not only nucleus, when he observed a cells of these archaea, then he observed what? He observed the other three, means in that cells only, he observed another structure, and he given the structure's name as stomata. Understand? That means the first stomata was also seen by Robert Brown. First name nucleus is also given by, discovered by Robert Brown. So here in which type of cell leaves he observed? The cells of orchid leaves. Orchid leaves. Understand, Anna? So in the leaves, along with the nucleus, he observed another structure, and he given the name of stomata, and he also given the function. It is helpful for exchange of gases. Exchange of gases. So all the students mainly focus, even in your 7th and 6th also, you people have come across, even in your 5th also, you people have come across about the stomata, part plants and functions of the plant. So here the discovered by stomata was first seen in orchid leaves by Robert Brown along with the nucleus. So once again, I want to repeat that the significant observations of to understand about the cell. Why? What is the need of its contribution? To understand about the cells and what are the cell organelles are prepared. So by so many scientists have showed an interest, and they want to know about what are the integral, means inside what are the parts are present and what is the importance of the part. So many scientists, they started knowing about, they started discovering. Understand? So in your 8th class especially, you have to focus only one thing in your brain, that here who is the person who observed the nucleus in epithelial cell. Epithelial cells are the outermost layer of the animal body. Again I am saying outermost layer of the animal body. Understand? Outermost layer of the animal body was first observed by Phyllis Fontana in 1730. That is, it is in each year. And then later, the Robert Brown, he observed all these structures. How they will observe? Through microscope only. So here he observed different kinds of cells. Understand? Different kinds of cells. Among them, that means he observed epithelial cells, so many cells. Among them, he observed in the cells of arcade leaves, he observed circular spot and more opaque. That means you can see very clearly that structure. Understand? And he said that it is an integral part of the cells, and it is a integral part and very important part of the cell, and it is also called as the cells control room. Cells control room. Even the cells control room is not given also. These words you have to keep in your mind, because in 8th class they are asking very important, which is called as cells control room. And who said integral, that is a part of the cell? Who said that? Robert Brown, who is the first person seen the first time in epithelial cells, that is the Willis Fontana. What is epithelial cells? It is the outermost layer of the animal body, outermost layer of the animal body. So why? Because when the CCE exam pattern is going on, every word in the textbook is very important. So here not only the nucleus, but Robert Brown, he observed another structures along with these, and he given the structure name as stomata. And stomata are very important, and he said what is the function of the stomata is the exchange of gases. Exchange of gases. Understand? So here in then next we are going to learn another activity, observing of nucleus in onion peel cell. So here what we have learned about about Robert Brown and what type of cells he has seen, and he given what the name to that cell organelle we have seen. Now here we are going to observe observation of the nucleus in the onion peel cell. Onion peel cell. Here we are going to see a comparison between the cells of cheek, understand, and the onion peel. Even we can see both the nucleus in that. Then what are the differences you are finding in both cells? We are going to discuss. Okay, Nana? So I hope everyone have understood about the scientist and how they have observed and what he said, everything. Here, then what is our aim? So in this activity I already said that everyone in a lab activity in regarding CCE questions, these type of questions generally they will ask. So that is the reason every student should know what is the aim, what are the apparatus required, what are the steps involved in the procedure, what is the conclusion, what are the precautions. So these type of questions directly the model is not given, but in CCE definitely in examination, so these type of questions will be given. So not to get trouble, I am explaining these experiment in regarding to the CCE model. So let us see, Nana. So what is our aim? Our aim is to observe. What, Nana? To observe nucleus in onion peel. In onion peel. Understand? So we are observing the nucleus in onion peel cells. Onion peel cells. Then apparatus. What are apparatus required? So apparatus required. I already said it previous. So they may give apparatus and they will ask what is the activity by the given apparatus. Which activity can we do? So please everyone keep in your mind. That means the apparatus are, what are the apparatus we require? We require onion peel. So this is the onion peel. 100. drawing please everyone. So I am taking an onion peel. We need glass slide. So I am drawing a diagram of glass slide. We need cover slip. So already I am shown I am drawing. Then we need water. So mug of water we are taking. We need glycerin. What is the need of glycerin? Here not to dry the material which is present in the apparatus, on the glass slide. Then here we need a stain. We need saffron. Saffron. We need methylene blue instead of that one. Methylene blue or red ink. Then we can take R instead of this or this. Then we need a blunt needle. We need blotting papers. Or these are used to take excessive strain or excessive water. Then we need microscope. Then these are the materials required. So we need onion peel, we need a glass slide, we need a cover slip, we need water, and we need dropper also. Now I will show you dropper. Understand? Then we need glycerin not to dry the slide. We need saffron, stains of saffron, methylene blue and red ink. We need stains of saffron and methylene blue. Stains of saffron, methylene blue and red ink. And we need a blunt needle. Understand? Means you see this is sharpened. We can use the blunt end of the needle to tap it. So we need a blotting paper. Blotting paper we can take for drying the excessive water from the glass slide. We need microscope. We need dropper. So please everyone, if they will give these, and I want to tell what is the need of saffron, methylene blue and red ink. If you are not having saffron, you can use methylene. If you are not having red ink, you can use saffron. So these are used to stain the nucleus. It is used to stain the nucleus. Understand? To stain the nucleus. So again I want to repeat. We need onion peel. I already said how we will take onion peel. Take the onion bulb, cut one portion, break it, then you will get a translucent layer. This translucent layer, understand? Then take the translucent layer and this is the onion peel. Then we need a glass slide. We have to place these on the glass slide. And we need a cover slip, water, glycerin, stain, either saffron, either methylene blue, either red ink. Then we need blunt needle and we need blotting paper to take away the excessive water from the glass slide. We need microscope. We need dropper to put one drop of water. Understand? So these are the materials required. How they will ask the questions. And I will explain what are the questions generally being asked. Generally being asked by the examiner. Understand? So I hope everyone understood what are the materials required. The aim to observe the nucleus in which cell, onion peel cell. These are the materials required. Okay, now we will go for the procedure. We will go for the procedure. So here what is the procedure? I want to tell you stepwise, everyone. Please see, because these steps can be given in a jumble manner and they will ask to write which step will come out other one after other. So first one, take onion peel. So we have to collect the onion peel. Place it on glass slide. Where we have to place it? On glass slide. Then put a drop of water. Okay? Put a drop of water. Then add glycerin. Add glycerin. Then add the stain. What stain? Saffron. If not there, methylene blue. If not there, red ink. You can use any of these three. Understand? R. I am writing very clearly R. So then after adding, cover with the cover slip. Then leave it for 5 minutes. Leave it for 5 minutes. So I want to write here. Leave it for 5 minutes. 5 minutes due time. Leave it for 5 minutes. After 5 minutes, understand? After 5 minutes, this is the, I am drawing here, you can see very clearly. Slide. You placed the material over here. Then you put all the drops of water, glycerin, then stain, then cover slip. Okay, this is the position. So then later leave it for 5 minutes. Leave it for 5 minutes. Leave it for 5 minutes. After 5 minutes, slowly put one drop of water from the sides of the glass slide. Means from here you will keep one drop. Understand? Why? To take away the excessive strain or to drain the excessive strain. Understand? Then what you have done? First leave it for 5 minutes. Then put a drop of water one by one. Understand? One drop of the water with a dropper. Then what is the need to use the water? To drain the excessive strain, and to drain the excessive strain, here we are taking blotting paper. Understand now? What is the use of blotting paper? The blotting paper will take, means one side if you are putting the drops of water and another side you have to apply you have to keep the blotting paper. What will happen? It will take away the excessive strain. Why? Because if excessive strain is there, you cannot see clearly the slide. So here if we are taking the excessive to remove the excessive strain, we are using water. On the other side, we are keeping the completely blotting paper or any that is, we will take filter paper or anything, then we will absorb, it will absorb completely the excessive water. Understand? So from the other side you are using a filter paper, a blotting paper, here you are using a one drop of water. So what is the use of water? Water will take away, will remove the excessive strain, and the removed excessive strain stain is being dried by the blotting paper. Understand? So for every apparatus, definitely the usage will be there. Understand? Usage will be there. Then here, after removing everything, take a blind end and tap it very slowly. Then why? Because the any air bubbles are there, it will be cleared, and place this under the microscope. So once again, I want to explain this one. Everyone, please clearly take onion peel, place it on glass slide. Put a drop of water, add glycerin, then here add the strain, either it should be saffron or methylene blue or the red ink. And saffron, methylene blue or red ink. I am saying R. Instead of this. This is helping to stain the nucleus, and easily you can see the nucleus clearly in the cell. So again I am telling the function. This saffron, methylene blue, red ink or a stain. It will stain. Stain itself will say that it will. These stains absorbed by the nucleus, and you can see clearly. Then here leave it for 5 minutes. Put a drop of water on from the side of the glass. Side of the glass means if the glass is there, you are using one drop of water. Then from the other side, place the blotting paper. Place the blotting paper. Understand? This function of the blotting paper, it will absorb all the excessive water which is coming out and excessive strain which is coming out from the slide. Then here after dry, then put them, tap over it not to have any air bubbles, then see under the microscope. When you observe under the microscope, you can see these structures. I will draw in a neat manner, don't worry. Just 2 cells I am drawing. So you can see because I use it as a so you can observe this is the cytoplasm. You are observing a clear structure of cell wall, and you are seeing a circular spot which is not in center. Please listen my version, which is not in center. It is called nucleus, and the part is the fluid part which is present is cytoplasm. So take onion peel, place it on a glass slide, put a drop of water, add glycerin and add strain, cover with the cover slip, leave it for 5 minutes, and after 5 minutes add one drop of water from the side of the glass slide, and another side place the blotting paper, and here then you can after drying put them under the microscope. When you observe under the microscope, you can find these type of structures which I want to show you in the book. Please, you can see very clearly this stain, this one, the circular bodies, blue color circular bodies will be present. You can see completely the cell structure, the outer layer you can see very clearly which gives the shape. Understand? They are arranged compactly. You can see a circular blue color dense body which is not in center in all the cells. This is called as nucleus. So what precautions can we take? See, we have to take very careful while you are adding the stain. You should not add excessive stain. Understand? While drying also you have to place the blotting paper very clearly. Understand? So I hope you everyone understood the experiment, the activity, how to observe the nucleus in onion peel cell. In the next coming video, we are going to see the observation of the cells, that is observing the cheek cells in the human being. Thank you. I hope everyone understood the video.